Hello, fellow biologists. This is Professor Tarun once again. I'm going to provide a couple of lecture videos on the next segment of Unit 4, and this covers Section 4.2 of Chapter 4 on translation. This follows the lecture on transcription, which centers or focus on the central dogma. We started actually covering this in detail, beginning with the discussions on DNA replication at the end of towards the end of Unit 3. And so the first part of Unit 4 started with the second aspect or the second level of the central dogma, which is transcription. And now we finally come to the third and final level of the central dogma, which is translation. So here we are going to see the underlying mechanism by which proteins are synthesized inside the cell. The core concept of translation is that it is a process in which the sequence of bases in messenger RNA or mRNA specifies the order of successive amino acids in a newly synthesized protein. We know in Unit 1 that proteins are linear polymers of amino acids that form three-dimensional structure with specific functions. So it is the sequence of amino acids, the successive sequence of amino acids that ultimately determine the function of the proteins. And in translation, we will understand how the successive amino acid assembly in order to produce newly synthesized proteins is actually directed by the sequences of bases in the messenger RNA. Your only reading assignment for Chapter 4 is Section 4.2. So please take note of this. Don't worry about Sections 4.1, which we in fact already covered in Unit 1, or the subsequent sections, or Section 4.3, and so on, because we are not going to cover those sections at all. So please take note that here in Unit 4, your reading assignment is only Section 4.2 on protein synthesis, and they are found in pages 80 through 86 in your textbook. This is a very dense section, however, so I would like you to pay attention once again to the various objectives under the learning objective of 4.2. So let's go through them individually. So the broad learning objective of 4.2 is to discuss the process of translation by which the sequence of bases in mRNA specifies the order of successive amino acids in a newly synthesized polypeptide. So the sub-objectives would be for you to be able to explain the roles of the major cellular components that are necessary for the process of translation. 2, to explain the role of ribosomes in the process of translation. 3, to explain why the mRNA transcript of a protein coding gene has 3 possible reading frames and how the correct reading frame is established in the cell. 4, to relate the structure of tRNA to the process of translation. 5, to present the argument that amino acyl tRNA synthetases are directly responsible, this is very important by the way, for translating the codon sequence in mRNA into a specific sequence of amino acids in a polypeptide. This objective here frames the basis for what we call as the genetic code, as we will see more closely. 6, explain how experimental data using synthetic RNA molecule made breaking the genetic code possible. We will gloss over this objective actually, so we probably may not have the time to go over this. 7, describe the main properties of the genetic code. And 8, to describe in detail the 3 steps of translation. So the last two objectives are the substance of part 2 of this lecture video. Okay, so we will focus on the other earlier learning objectives for this particular video. To assess your understanding of the section, so please try to spend time answering these self-assessment questions. First, what are the relationships among the template strand of DNA, the codons in mRNA, anticodons in tRNA, and amino acids? 7, or number 2, which polypeptide sequences would you expect to result from a synthetic mRNA with the repeating sequence that is indicated here? 8, what are the steps of translation? Name and describe each one. And 9, bacterial DNA containing an operon encoding 3 enzymes is introduced into chromosomal DNA in yeast, which is a eukaryote, in such a way that it is properly flanked by a promoter and a transcriptional terminator. The bacterial DNA is transcribed and the RNA correctly processed, but only the protein nearest the promoter is produced. Question is, why? This question is actually very important in terms of you guys integrating your understanding of transcription in the previous lecture videos by Professor Bosch, and also your understanding of translation in this section. And please take note of the differences between transcription and translation in prokaryotes and transcription and translation in eukaryotes. So this question here, last question, number 9, would test your ability to integrate those key important understanding. Before we look at the translation more closely, I would like to review the central dogma with you in order to provide us with a proper context to understand translation better. You were introduced to the idea of the central dogma in Unit 1, where you realize that central dogma is simply a description of the way genetic information is transmitted or flows inside the cell. And the classic central dogma, or the traditional view of central dogma, simply describes how information flows from DNA into messenger RNA by the process of transcription, which in detail you have already seen in Chapter 3, the first part of Unit 4. And then from mRNA into proteins through a process called translation. So in transcription, the sequence of bases along part of a DNA strand is used as a template. In other words, a small segment of the DNA is used as a template in order to produce a complementary copy in the form of a messenger RNA. And this messenger RNA encodes the sequence for a protein, so that in translation, the sequence of bases in messenger RNA is used to specify the order in which successive amino acids are added to a newly synthesized polypeptide chain. So translation, therefore, is the last stage of the central dogma of molecular biology. So this is the central dogma in its simplest form, in the most traditional form, but throughout the years, the central dogma has gotten more complicated, And in fact, in a series of chapters that we have already encountered, we saw some of the additional complications or varieties by which um, genetic information can flow inside the cell. And one of these examples is when we encountered with viruses through the video lectures of given by Professor Bosch in chapter 13 is the case where an RNA, for example, can be used as a template in order to generate a DNA copy by a process called reverse transcription. Okay. Also, we have seen in chapter 3 that DNA can transmit its information into new daughter strands or daughter DNA molecules through the process of replication. And then, of course, there are other viruses, like the COVID-19 viruses, where the mRNA can actually generate more copies of itself, right, through the process of RNA replication, okay, using very special types of RNA polymerases, okay. The only thing that we haven't seen happening so far, we have no proof or any demonstration of it happening in nature, is the reverse information flow from the amino acid sequence of proteins to mRNA or DNA. So this process we haven't seen thus far. Okay, so this is the central dogma in its modern view because of the discoveries that we have seen in many other phenomena, including, of course, viruses. It is important also to remember that this so-called central dogma happens only in 2% of the entire human genome, as we saw in chapter 13. Okay. So what about the rest, the 98%? Well, about 90% of these, through the Human Genome Project, has been shown to produce what we call as non-coding RNAs, or ncRNAs, okay, whose function it is to regulate the process whereby the central dogma happens. The flow of information from DNA to protein is regulated by these non-coding RNAs. And in fact, when we go to translation, we will see examples of non-coding RNAs such as transfer RNAs and ribosomal RNAs. But there are many other types, new classifications or new classes of non-coding RNAs that we won't cover in this course, but it is important for you to have this taste or general insight into what is happening in the broadest, most current view or understanding of the central dogma and its relationship to the rest of the human genome. So the important point to remember then is that the central dogma is not the predominant manner by which information flows inside the cell, but it is central because it shows the way by which mRNAs, that codes for proteins, gets expressed. But the control by which these proteins gets expressed, when and where inside the cell, that makes us the complex organism that we are essentially dominated by the regulation of um, this process through non-coding RNAs. So in this video, which I call as part A of section 4.2, we will have a closer examination first of the molecular machine and its working parts. Translation of proteins is probably considered as the most sophisticated um, uh, process involving a very complex molecular machine in order to undertake the process of synthesizing proteins. So let's have a close look at the working parts of this wonderful machine. The translation process is a very complex process that requires multiple components in order to successfully occur or take place. And we can, of course, get a little bit of a feel or a sense of the complexity of this process by looking at the key components of this complex molecular machine. And so there are about nearly 100 different types of working components that have to work in harmoniously and cooperatively and in a sequential order in order for a newly synthesized protein to be manufactured. And so these different components can be categorized into several groups. The first, of course, is the messenger RNA. It is illustrated here as a naked RNA, but in reality, these messenger RNA are bound by RNA binding proteins. RNA binding proteins. So the RNA, there is never an RNA that is naked inside the cell. It's always covered by RNA binding proteins. And there is the what we call this initiation factors. These are important components that prepare the cells for the initiation stage of translation. There are also elongation factors that are responsible for allowing the process of translation to speed up and happen in a directional manner. And then there is the release factors that are responsible for terminating the process of translation by recognizing specific signals. There are the critical family of enzymes called amino acyl tRNA synthetases, which are responsible for adding the specific amino acids in the special type of RNA called transfer RNA. So these enzymes here are responsible for adding the specific amino acids that gets attached to the transfer RNA. And of course, there is this mega machine. We call these mega machines because it's actually made up of about three different types of RNA called ribosomal RNA, okay, plus about up to greater than 50 different types of ribosomal proteins. Okay, or R proteins. Okay. So these are the uh, parts list of the incredible molecular machine that is responsible for synthesizing proteins. So let's have a closer look at how or what this each of these categories do and when they do their function. So let's have a close look at the central mega machine that is responsible for the synthesis of new proteins. The ribosomes are appropriately called the protein factories, where translation takes place, because they are the ones that are actually responsible in coordinating all of these complex process and catalyzing and decoding the assembly of the correct sequence of amino acids in order to build um, new polypeptides. And as we have already alluded to earlier, ribosomes are a highly complex structure of ribosomal RNAs and ribosomal proteins that binds with mRNA. The ribosomes consist of a small subunit and a large subunit. So why is this so? The small subunit is responsible for what I call as the decoding function, the decoding function of the ribosome. And when we say decoding, it is responsible for um, coordinating the interaction between the sequence in the messenger RNA and the sequence of nucleotides in the tRNA carrying an amino acid, and therefore provides the way by which these amino acids are assembled in sequence. That's why it's called decoding. Okay. The large subunit, on the other hand, is responsible for the catalysis, the chemistry aspect of the assembly process, where what we call as the peptide bond formation is happening. Okay, so this is the main role of the large subunit. So the small subunit is responsible for the decoding of the messenger RNA, and the large subunit is responsible for the formation of the polypeptide bond. Okay, so in eukaryotes, the ribosomes are much larger than prokaryotic ribosomes, understandably, because they require more regulation. The central mystery of the mechanism of translation is the question of how the two-dimensional sequence of nucleotides in a messenger RNA can actually be translated into the three-dimensional structure of a protein. In other words, how does the ribosome decode this linear sequence of nucleotides in messenger RNA into an assembly of a protein that has a very specific function in three-dimensional world. In fact, this is what drew me into this phenomenon as a scientist. So, in fact, my research in my PhD degree is in trying to understand on how this process happens. So, the key um, um, aspect that unlocks this process is so-called the reading frame. So what we need to understand is that in an mRNA, you have a series of sequence, let's say UA, G, C, A, U, A, U, G. And the question is, how does the ribosome know when to start reading the sequence? And in what way is it reading it? And so the discovery was made that the ribosome actually reads the sequence into three-letter words called codon. Okay. The question though is, when does it know how to start? Okay, so there are three what we call possibilities of reading frames. It can either start in the first nucleotide, okay, and then reads three each, like so, etc. or it can read in the second. So nothing necessarily prevents it from doing so, or the third. So this is what we call as the three possible reading frames whereby a ribosome can start reading the sequence of nucleotides in order to assemble amino acid uh, uh, sequences into polypeptides. The other is what determines where to start. So it's always possible to start at any point in the sequence. So, in order to understand this mystery, we have to look at more closely at how the large subunit and the small subunit coordinate together in order to establish what we call as the correct reading frame for the codons. The detail of how this is discovered is actually in how do we know section of your reading assignment. We won't have a chance to cover this, but if you're really interested in going into detail, feel free to read that. It's a beautiful, elegant series of experiments that won the people or the scientists who discovered them a Nobel Prize. The large subunit of the ribosomes includes first three binding sites for molecules of transfer RNA. This is called the A site, okay? And this is what we call as the amino acyl site. And we will see shortly what that means. And then there is another, the middle part is called the peptidyl site. The P site is where the catalysis actually happens, formation of peptide bond. And then E is the exit site, where the tRNA that has been previously used gets released, okay, or exits out of the process. And so these three main sites are the ones responsible in coordinating the process of catalyzing the peptide bond formation. Now, the ribosome, particularly the small subunit, together with a very special type of tRNA called the initiator tRNA, which carries methionine. And this is the universal attribute of the initiator tRNA, is that it carries a methionine with it. This is the one that is responsible or that determines the correct reading frame for the codons. So I already indicated that the codon is in each group is each is each a group of three adjacent nucleotides coding for a single amino acid. So one codon correspond to one amino acid, okay? And a codon is made up of three nucleotides. Okay, so with respect to the codon, the words therefore are made up of simply three alphabets. The alphabet corresponds to the nucleotides. So each of these codon corresponds to one type of amino acid, one kind. And the reading frame is where the ribosome begins reading the sequence in the mRNA. And so we will see shortly how the initiator tRNA in coordination with the small subunit does that. And the most important rule that you need to remember is that it's the first AUG, this sequence here, this codon, AUG codon, marks the beginning of the correct reading frame relative to the 5' end. So in this sequence that I've written here, the ribosome can actually scan this and look and look and look and see whether it can encounter an AUG sequence. And what we see here is there is an AUG sequence right here. So this is considered the first AUG sequence. So in principle, this is where the ribosome starts translating. So this is where the reading frame begins. Now let's have a look at tRNA. we can appreciate the function of the tRNA if we also have a proper understanding of its structure. So this goes back to the theme of tRNA, the structure of tRNA defining its function. Okay, so the transfer RNA molecules are, as we have already seen in the previous slides, are key to establishing um, the correct reading frame, but it's also responsible for carrying the right amino acids to deliver for assembly. Okay, so that's why the transfer RNA has two important business ends. The first is the amino acid attachment site, which is found at the 3' end of the tRNA molecule. And then there is another structure called the anticodon loop. Now, the reason why it's called a loop is that if you notice, the tRNA, which is actually a very common attribute of RNAs, is they exist as single strands, but inside the cell, these single strands can actually fold back upon itself in order to form what we call as secondary structures. So in this particular case, imagine this is a single molecule of tRNA, and yet it folds back upon itself in order to show regions. So this is the 5' end, okay, folding back, and then again folding back like so. And so there are regions that are double stranded, such as this. These are called stems because they look like stems. So this is the duplex regions where there is complementarity of base pairing, also here. But where there are also regions that don't have any kind of uh, complementary sequence, so they form single-stranded region or loops. So whenever we speak of loops, it indicates a single-stranded region in an RNA molecule. And so the anticodon, therefore, is called an anticodon loop because it is a region where the molecule is single-stranded. And the anticodon is complementary to a codon in an mRNA. So that is the key to the role of the tRNAs. Okay, and if you look at the actual three-dimensional structure of tRNAs in solution, X-ray crystallography has shown that it actually forms what we call as a looks like a clover leaf structure. So it has a three-dimensional shape, a very highly structured architecture that is actually very similar to what we see in proteins. Okay, so tRNAs therefore are single-stranded RNA molecule that folds back upon itself to form secondary structure, which then forms higher order three-dimensional structure in solution. And this is what makes the tRNA very specific in its function. In other words, there are many different types of tRNAs that correspond to different types of amino acids. Okay, so again, I would like to emphasize in the three-dimensional structure, you notice here that the anticodon region, the anticodon loop, actually has three nucleotides that are exposed in solution that are ready to interact with a codon in an mRNA. Whereas the single-stranded region at the 3' end, okay, is a key to the attachment of a specific amino acid. Now let's look at tRNA synthetases and the important function that they play in translation. So these tRNA synthetases are actually protein enzymes. They connect the specific amino acids to specific tRNA molecules, as shown here in this figure. It's, it's important to distinguish a tRNA that has amino acids with tRNAs that have or that carry amino acids. The tRNA without an amino acid attached is called uncharged tRNA. Okay. Now, if the amino acid is attached to it, then it is charged. So it is the responsibility of tRNA synthetases to charge the tRNA with a specific amino acid. And how does it do it? Well, if you notice here, what you have is that each amino acyl tRNA synthase have a specific binding site for a particular tRNA and also a particular amino acid. So it is therefore the tRNA synthetases that determine which amino acid gets attached to a particular uncharged tRNA. So the specificity also therefore of the amino of the enzyme, okay, in this particular reaction is therefore the basis for the what we call as the genetic code in cooperation with the complementary codon anticodon interactions between tRNA and mRNA. Consistent with this key principle, there is in fact a specific enzyme, specific tRNA synthetase for each of the amino acids. And in fact, these tRNA synthetases are very accurate and they rarely attach the wrong amino acids. Although occasionally some mutations can happen that allows the misincorporation or attachment of the wrong amino acid to tRNA, which leads to a devastating disease states. So the previous slide emphasizes the role of tRNA synthetases in determining the basis of the genetic code because of the specificity of this enzyme in attaching um, the right amino acid to the right tRNA. And so that is a crucial part of the genetic code. But the second aspect of the genetic code is what we call as the codon anticodon interaction, which is now the interaction of the tRNA itself through its anticodon region with the particular codon in the messenger RNA as shown in this figure. Now, when we speak of codon anticodon interactions, it does not exist in such naked fashion, but always think of the context of this process in relation to the small ribosomal subunit. Oh, I'm sorry. Ribosomal subunit. Okay. In other words, this process of codon anticodon interaction, not depicted here, but as we will see shortly, okay, happens through the on the surface of the ribosomal subunit, because the messenger RNA has to be bound by the small ribosomal subunit in order for this codon anticodon interaction to happen. So please take note of that. Make sure that you have that in your mind as you go through this process. So in this figure, what it shows you is, of course, first the double-stranded DNA. It's actually demonstrating again the central dogma, where the template strand, which is what we call as the negative strand, generates a positive strand mRNA that codes for the protein, and then that positive strand mRNA gets translated. So the specificity of the DNA RNA as well as codon anticodon interaction uh, results from what we call as complementary base pairing. So this is always a rule in genetic information transfer. So when we see what happens during the process of translation, we see one codon in the mRNA. Okay, and notice the orientation of the mRNA here: 5 prime, 3 prime. Okay, and the sequence of the codon is AUG, right? 5 prime, 3 prime. This codon then interacts with the initiator tRNA. So imagine that this is the first AUG that gets encountered by the small ribosomal subunit. And when it does so, it then allows its bound initiator tRNA to recognize the AUG via the initiator tRNA's anticodon. So this is the codon, and in the mRNA, it's AUG. Now take note the complementary base pairing. Uh, the complementary base pairing then would be from the 3 prime to 5 prime of the anticodon is going to be UAC 3 prime 5 prime. Okay, so please take note the again the anti-parallel nature of the interaction between the codon and the anticodon. It has to be anti-parallel. So this forms the basis for recognizing the correct codon in incorporating the first amino acid, methionine, during the process of translation. So to emphasize the anti-parallel nature of the base pairing between codon and anticodon during the process of decoding, the consequence of that would be that, as indicated here in main point number 1, is that the first base in the codon, so this is the codon, right? The first base is A from the beginning from the 5 prime. The first base in the codon will therefore correspond to the last base of the anticodon. So 3 prime U A C. So this is the codon. This is the anticodon. Okay. Now, most codons specify an amino acid according to a genetic code as a result of this combination of tRNA synthetases charging a specific tRNA with a specific amino acid, and then the codon part of that specific tRNA recognizing a specific codon. Now, again, it's important to remember that the codon that initiates the process of translation is always AUG, which corresponds to the amino acid methionine. So that is a key important point to remember. Now we end this video with the uh, so-called standard genetic code, which is the holy grail of molecular biology. So in other words, this is the pattern that we expect to see to be able to decipher how sequences in nucleotides gets translated into sequences of amino acids in building proteins. So I expect you to actually have a working knowledge of the genetic code. In other words, how to use them by looking at the codon. I would expect you to be able to decipher what it means in terms of the amino acid that is coded for in the protein. Okay, so the way to use it, and I'm pretty sure most of you must already know how to use this, is to first look at the first position of the nucleotide in a given codon beginning at the 5' end of that codon. So this is the reference point, therefore, here in the code is that this is a sequence of the codon from 5' to 3'. So imagine yourself to be the ribosome trying to look at the run of sequences in the messenger RNA and then looking at one codon at a time. So, for example, if the first codon would be has U at the first position, and then in the second position of that codon, it also has a U, and in the third position of that codon, it also has a U. Okay, so that means to say that you need to look at where these 3 points converge, and what you have is that UUU codes for phenylalanine. Okay, another example would be, let's have a look at that methionine that we have seen before. So methionine is codon is 5' AUG 3'. So the first amino acid, methionine, in any protein that gets started is coded by AUG. So the first position is A. So this is A. The second position is U. So where is U? So this is U also. Okay. And then the third position is G. So if we look at where those 3 points converge, it converges at methionine. Okay. So if we look at the overall pattern in the standard genetic code, in fact, there are 64 total different codons. So you count each of the combinations of these letters in this table, and you get 64 different codons. But if you look carefully, only 61 out of these codes for the 20 amino acids that we know constitutes the different types of amino acids in proteins. Okay? In other words, many amino acids are specified by more than one codon, which therefore makes the genetic code what we call as redundant or degenerate. What do we mean by this? For example, if you look at the codons that code for phenylalanine, PHE, this is phenylalanine, that there is actually 2 different codons that can code for it. These are called synonymous codons. Synonymous. Just like there are same words that express the same meaning, there are also different codons that can code for the same amino acid. So that is the concept of redundancy or degeneracy. Redundant or degenerate. Okay. You can also notice that there are codons. Remember, we said that there are 64 different codons. So what happened to the other 3? The other 3 actually does not code for any amino acid. Instead, they are used by the ribosomes to interpret that translation have to stop once it encounters any of these 3 codons. So these are called stop codons. Okay? And the other color code simply refers to the chemical nature of the different amino acids. So some of these amino acids, as we have encountered in Unit 1, would be nonpolar amino acids. Some would be polar. Some would be basic. Some would be acidic. And of course, the three codons that represent the stop codons. So in summary, this is the standard genetic code. And so by try to practice looking at this code and try to identify amino acids corresponding to their different codons. Okay. So to end this part A video, please answer this brief question. Because there are three different possible reading frames in a messenger RNA or mRNA molecule, most mRNAs can be translated in a cell into three different proteins. Is this true or is this false? So please consider again the principle of how ribosomes establish reading frames to answer this question. Okay, so our next video is going to be part B, and that should complete the unit segment of chapter 4. Thank you.